This is a series of several short presentations on Helicobacter pylori and the diseases that it causes. So let's start off with a little about the organism and its discovery. There's an interesting story there. So generally, the bacterium is described as a flagellated spiral gram-negative rod. And so from the diagram on the left, you can see that this particular specimen, shown in a transmission electron micrograph, looks a little vibrio-shaped. And we'll come back to the shape of the organism in a little bit, but generally it's described as a spiral gram-negative rod. And the bacteria was discovered in 1983 in the stomachs of patients with type B gastritis. Now, the bacteria was known before this, but this is when interest grew in the organism. And so it's discovered in the stomachs of patients with type B gastritis. And that is chronic inflammation of the stomach antrum. And for those of you that are anatomically minded, that is the region of the stomach that is just before the pyloric sphincter. And the pyloric sphincter is the ring of muscle which controls the exit into the duodenum, or I think as Americans like to say, the duodenum. So this bacteria was originally classified as a Campylobacter species, but it was subsequently reclassified as Helicobacter, and the specific species we are interested in is the Pylori species, so we're talking about Helicobacter pylori. So this organism has now been clinically associated with gastritis, with peptic ulcers, with gastric adenocarcinoma, and with gastric malt B-cell lymphomas, and we'll talk about all of those in more detail a little later. Let's take a look at the top right image here. The top right image here shows a biopsy of gastric mucosal epithelia from the pyloric antrum of Dr. Barry Marshall. And Dr. Marshall had 8 days earlier drank a culture of Helicobacter pylori. So obviously something we would never ever encourage anybody to do in lab, but Dr. Marshall had a very motivating reason to do this. He and his colleague Robin Warren had for a number of years suspected that Helicobacter pylori was the causative agent of stomach ulcers. And if you're familiar with stomach ulcers, these have often been attributed to acid production in the stomach or things like stress and anxiety. But after Dr. Marshall drank his culture of Helicobacter pylori, he subsequently went on to demonstrate that the organism is associated with these various gastric diseases. And so in this top right-hand image, you can see that there are these pits, and that's a gastric pit in the lining of the stomach, and down here this is a pit that we're looking down on top of, or we're seeing cut in section, and so then these little clusters of what look like spirochete bacteria are groups of Helicobacter pylori, which are colonizing the gastric mucosa, and that's the layer of mucus that lines the stomach. So this is a biopsy from the stomach of Dr. Marshall, and so they were able to show that this pathogen is able to colonize the stomach mucosa and colonize the gastric pits, and is causative for gastric ulcers, other peptic ulcers, and that range of diseases I mentioned earlier. So let's now take a look at the physiology and the structure. And like I mentioned, it's a gram-negative spiral rod-shaped bacteria. But in culture, the bacterium is pleomorphic, and that means it can adopt a variety of shapes. So if we look down here in the bottom right, you can see that that's a good example of a spiral-shaped bacteria. But then there are some which this fella here is clearly coccoid, and some of them are more vibrio-shaped. And so these are pleomorphic, meaning many different forms. And so this depends on the age of the culture, the nutritional conditions, and that kind of thing. And generally, as you get older and older and older in the culture, you see more pleomorphy and you see more coccoid forms. So that's the first point there. The The bacteria is classified by a number of molecular criteria, and so the primary one here is 16S ribosomal DNA typing. But species and subspecies can also be characterized by their fatty acid content and by the possession and the arrangement of flagella. So those are some molecular criteria, and clinically some species and strain information is obtained from the specific site of colonization, and some species will actually colonize the duodenum, and others are restricted to the pyloric antrum, and others have been identified in the cardiac antrum. That's the cavity of the stomach towards the top of the stomach, which is near the sphincter of muscle that controls the entrance into the stomach. So then on to point 3, the bacteria, and this is very important, the bacteria possess a number of polar flagella. So that just means they're at one end. If you think back to your bacterial structural material, that just means that there are numerous flagella stuck to one end. And that makes the organism highly motile, and that's going to become very important when we talk about the pathogenesis, that is how the organism causes disease. And so bottom left here is a negatively stained image of Helicobacter pylori, and you can see the numerous flagella that are present on one end of the bacterium. So that's what you're looking at in the bottom left. So the flagella are important because they're a determinant of virulence, because they allow the bacteria to swim through the stomach and burrow through the mucosal lining to get to the epithelial surface in the stomach. And we'll talk about that more a little bit later, and why that's a specific virulence factor. The enzyme urease is produced by the bacteria. This is specific for the species which infect the stomach. And the reason that this bacterium will produce urease is that it creates a buffering environment where the effects of the acidic pH of the stomach are felt by the bacterium. So we'll talk about urease production in a little more detail in a moment, but that is also a virulence factor for the species which colonize the stomach. Now, if you're a species which colonize the duodenum, then you don't need to worry about producing urease because you don't have to overcome the effects of stomach acid. So that's an important differentiating factor between the different types of pylori which infect different locations in the GI tract. And point 5 here, bacteria has been difficult to study, which is one of the things that hampered Marshall and Warren. It's got very complicated ex vivo growth requirements. So ex vivo just means out of the organism. So the media it's grown in has to be very, very rich. It's a complex media, and that's going to be media supplemented with things like blood, serum, charcoal, starch extracts, egg yolk, and the growth has to be microaerophilic because obviously there's not a lot of oxygen in the stomach and the duodenum, so the pathogen has to be grown under microaerophilic conditions, obviously at or close to 37 degrees Celsius, and so that kind of sums up some basic things about the physiology and the structure. Okay, the last slide in this little piece of the presentation is it's going to be the pathogenesis, and so that is how does the bacterium cause harm to the host. And remember, harm to the host is just a simple definition of disease. And so the way I like students to think about disease is a perturbation of a steady, stable state. And so a perturbation of a homeostatic mechanism will result in disease. And you can think about this from the context of diabetes. If you can't regulate blood sugar, then you get a drift from the normal homeostatic levels of glucose in the blood and in your cells, and that results in disease. And we know that disease is diabetes. So as we're talking about these, think about perturbations of normal, steady state conditions and how the pathogen perturbs those steady state conditions, because when they get perturbed, that's when you get disease. Okay, let's start off with one point about the stomach here. So point 1, the stomach contains about a half gallon of gastric juice, and that's pH 1 to 2. So that is not a fun place for anybody to live. It contains hydrochloric acid and it contains digestive enzymes, and they would normally destroy cells, and that's cells which are brought into your stomach from the external environment, so that come in on your food and in fluids, for example. But they'd also destroy the epithelia of the stomach, And the reason that ulcers are so painful and dangerous, but so painful, is that when you perforate that epithelia and you allow the stomach contents to seep through into the underlying tissues, then that's basically acid being poured onto, in some cases, highly innervated tissue, and that's going to be extremely painful. And then you can also then bleed into your stomach, or you can bleed into your duodenum, and that's very dangerous because that hemorrhaging is hard to stop. Okay, back to our stomach environment. So there's that mucosal layer which is going to protect the stomach epithelia from the acid in the gastric juice. And so that's where the bacterium is going to try and live. So it's going to live in the protective environment of the mucosal layer. Before it gets to the mucosal layer, though, it's got to survive the stomach acid. And so that's what this enzyme urease does. It basically takes compounds that contain urea, and that's going to be a byproduct of protein degradation. Obviously, there's a lot of amino groups in amino acids that are parts of proteins, and so as those proteins get digested, a certain amount of urea gets produced, and pylori breaks down the urea in the presence of acid. So the equation here of the urea plus the protons, obviously in an aqueous environment on the left of this equation, that is dealing with the breakdown of urea in the presence of acid and water, and that produces bicarbonate and ammonium. And the bicarbonate and ammonium act as a buffering system, and so they can control the pH around very close to the bacterium. So the urease is secreted, is kept very close to the cell surface. There's this debate about whether it's actually secreted or whether it is held on the cell surface, but we get a local buffering effect which allows the bacterium to temporarily survive the stomach acid while it is doing point 3, which is swimming into the mucus layer. And so that's the point of the flagella. And so the bacteria can move through the gastric juice, can burrow through the thick mucus, and thereby evade the negative effect of acidic environments. The mucus is very thick, and so that involves the production of an enzyme called, or requires the production of an enzyme called mucinase, or mucinase. And mucinase or mucinase breaks down mucus, so it breaks down the carbohydrates that are found in mucus, and that makes it easier for the bacteria to swim through the mucus layer. So also there in point 4, there are some other proteins which are considered to be virulence factors. And so the bacterium produces a phospholipase, which contributes to some epithelial cell damage. Phospholipases cleave the phosphate head off of phospholipids and thereby damage membranes. And so that's a virulence factor because it leads to host damage. There's a protein of newly emerging function called CAG-A, C-A-G-A, CAG-A. And CAG-A is, in some camps, thought to be an oncogene. So some people believe that Helicobacter pylori contains a protein which can directly stimulate tumorigenesis. And that's a little controversial right now, so we won't get into that. There's also a toxin called vacuolating cytotoxin, and that stimulates the production of many vacuoles in a cell, and that can lead to cell death. And so there are some virulence factors in point 4. And so think about why urease production, the presence of flagella, and then all of these proteins produced in point 4 are virulence factors. And that'll link you back to the content from last week. In terms of the actual disease, while there is some damage caused by the bacterium through the action of phospholipase and the vacuolating cytotoxin, most of the disease is believed to be caused by the host immune response. So there are generally 2 ways in which pathogenic microorganisms cause disease. On the one hand, they can produce toxins, and there's going to be exotoxins and endotoxins, that kind of thing. And on the other hand, they can cause an overwhelming host immune response. And so there are lots of diseases which are basically the manifestation of perturbation or loss of homeostasis in the immune response. And so you can think about a number of hepatitis disease-based diseases which are caused by inflammation. Some types of liver cancer are caused by chronic inflammation. Lots of examples of diseases caused by perturbation of the immune response, and that's mainly how we're going to look at the diseases. We won't look too much at the effects of phospholipase, CAG-A, and vacuolating cytotoxin. Another virulence factor that the bacterium has, which allows it to cause disease, is the presence of two enzymes, superoxide dismutase and catalase. And so when a pathogen invades a host and sets up a site of infection, so colonizes the host obviously has an immune response. And many pathogens work pretty hard to evade that immune response. And Pylori is one of those pathogens that tries to evade the immune response. And it does that partly by expression of superoxide dismutase and catalase. So when a pathogen is recognized by the immune system and it is attacked by phagocytes and the like, the pathogen can be engulfed into the pathogen and ends up in a phagosome, which is an engulfment of a phagocyte. And that phagosome is then bombarded with reactive oxygen and reactive nitrogen species. And the enzyme superoxide dismutase can convert some of those reactive oxygen species, things like O2 dot and other radicals, such as hydroxyl radicals, into hydrogen peroxide, H2O2. And then catalase can take that hydrogen peroxide and convert it into oxygen and water. And you've probably talked about catalase when you've talked about differences between strep and staph. And so that evasion of the immune system is a virulence factor, because if we take away the ability to evade the immune system can overcome the pathogen, and the pathogen no longer causes disease. So think about what are these virulence factors doing to cause disease, and ask yourself, if we take it away, do we lose the pathogenicity of the pathogen? Okay, so this is the end of this first part, and so that's 3 slides, and I will move on shortly in another video to some of the diseases caused by the pathogen.